Hi everybody, my name's Andy Fraser. I'm a professor here at U of T in the Faculty of Medicine in an institute called the Donnelly Center. And there we try and understand very large-scale genetics, essentially like how the human genome works and how our genomes dictate who we are and how our biology works. And one of the things I really want to get across to you today, since you're listening about what possibilities are ahead of you, is you are coming towards biology at really one of the most incredible times to be a biologist. And we all know that biology is incredibly diverse, but the unifying thing of all life on Earth is ultimately ACs, Gs, and Ts, the letters that make up our genetic code that dictates the way all the biology works to a large extent. And when I say that you are living a sort of an incredible time for biology, a lot of this is because of the speed at which we can read that information and the huge progress we've made in understanding it. So if I look at when I was a grad student, when I started, let's say 30 years ago-ish, one person could read about 10,000 letters in a day. And that was hard work. That's like a 10, 12-hour day to do that. Now you can read 10 billion a day easily. So that's a million-fold change in the speed at which we can get this information. And if you think about that in terms of travel distances and speeds, it took 4 days, 6 hours and 45 minutes for Apollo 11 to make it from Earth to the moon. 1,000,000 times faster, you'd get there in less than a second. If you could get to the moon in less than a second, you would all be exploring the solar system, right? You could zip to Mars within a couple of hours. It would be an incredible time to be a space explorer. That is the change in the speed that we have with biology. And what I'll give you is a couple of the very diverse ways in which that's changed the view of us as humans and in terms of the changes in the biology that are coming. And you're right at the stage where all of this is coming together. So one of the things that sequencing has completely transformed is our idea of who we are as a species. By sequencing many human genomes, and we've sequenced over 1 million human genomes by now, you can see how different genetic variants spread across the Earth, across different times. So we know almost exactly how humans spread across the planet now in incredible detail. We can also peer back across times, paleobiology. So we can look at Neanderthal remains, Denisovan remains, Homo erectus remains, and we can sequence those remains of ancient hominids. And we can see when Neanderthals inbred with modern humans, when these admixture events occurred. So we have ideas exactly about the diversity of Neanderthals, the diversity of ancient humans, all the crosses. We can identify new species, looking way back into the mists of human time. So this has totally changed our view of who we are as a species. It's also changed how we see ourselves. It's like we're not just humans, we're in a sort of roiling ecosystem, like a rainforest or something. We have hundreds of species of bacteria living in our gut, in our skin, between our teeth and our ears. 90% of the genomes in our body are not human, but we can see them, we can sequence them, we can start understanding how changes there affect our health and our behavior as humans, and how that all integrates into an entire ecosystem. And when we peer at human genomes, we find a lot of stuff that is not human. So when we purify DNA from humans before we sequence it, we carry a whole bunch of stuff along with that, including many, many viral genomes. And super smart computational biologists can stare at those data and pull those viral genomes back out of all the information that's dumped onto our computer servers. So mining these public data over the last few years has identified 100,000 new viral genomes. Like 100,000, just completely incredible explosion of information. And just to take one of the crazy viruses, we're at a new dawn of exploring these. So a mimivirus, and this is a picture of a mimivirus, obviously false colored a little bit, it's the size of a bacteria, right? That's an enormous virus. It has 1,000 genes. That's the size of a bacterial genome. And most of them, we have absolutely no idea what these genes do. And we're discovering these families of viruses all the time. Huge viruses, tiny viruses, all these different ecosystems. This is all exploding out. And it's all new information. It's already there for you to do PhD projects to figure this all out. The other thing that's changed is the precision with which we can manipulate and measure biology. So we can take a few cells from your blood, from your gut, from your skin, take those and turn them back into stem cells that can be used to make any other tissue that you want, just like the original cells in an embryo. And we can then take those stem cells and push them to form different types of cells in a test tube, right? And we can grow things that look like true organs in a tube. So what I'm showing you here is the gut, or follicles here. Those came from single cells. They grew them with particular conditions and grew things that look just like a gut. And because we can change now the sequence of the genome that came from the human to make the stem cells, we can look to see how individual changes of the genome would affect gut function, or would affect the ability of a drug to affect a cancerous gut. Incredible ability to regulate and change our genomes and look at biology. And ultimately, where that's all going is we're getting these huge collections of data, and we're learning faster and faster how to pull vast data sets together and make meaning from it. And all of you are aware about the huge changes that have been made with ChatGPT and other big LLMs. And we can now make incredible predictions, protein structures. We can predict how drugs will target proteins. We can predict many, many aspects. And so this marriage of huge amounts of sequence data, huge amounts of functional data, and computational power are opening up in biology in ways that honestly none of us dreamed 5 years ago. And you're right at the cusp of all of that. We can now write entire genomes. There are organisms that are growing in a lab where the whole genome has been made from scratch in a tube. You can make every single possible change and you can see what happens. So again, this systematic way of producing data, it's not one data point here and there. It's a huge cloud of data which we then use computers to make sense of. In terms of PhD itself, I just want to look at it in two different ways from what you'd experience if you go through a PhD here. And one is the purely professional one, and the other is a personal one. And these two things are both incredibly rich experiences as you go through a PhD. From the professional point of view, you get these incredible technical skills. You'll get to experience cutting-edge biotech. You'll understand how to wrangle vast data sets, how to code, how to use machine learning. Literally every grad student in my lab is way better than I am at everything they do in the lab. And not only will you leave a PhD as being a world expert in something, this will happen faster than you can possibly imagine. Within a few months, you'll go to your supervisor and say, hey, how do I do this thing? And your supervisor will say, I have no idea. You're going to have to go talk to that person. They might know, or go look it up online or figure it out. I have no idea. And the first time that happens, it feels terrifying, and then it feels super empowering because it's you. You're learning everything, and you're the person that's the expert. It's your project, and you're going to have to steer it. And those skills that you get, I mean, of course, they're useful in academia, and if you want to go on and become someone like me, good luck, another lab head. But that also lets you go into biotech, into the private sector. It lets you go into society and help you advise governments. So we have PhD students that have come out into both these things, biotech in startups or biotech in sort of major pharma. In terms of society, we have grad students that are now advising ministers for health, advising the Trudeau government into how to structure science and how to fund it and how to integrate it with healthcare and with IT. And then also, of course, there's the entire IP world where you need lawyers to help you own the bits of invention that you have and how to connect that with bankers and VC and finance. Those skills that you get in your PhD, you will be right at the forefront of being able to do any of those directions. And we have MoGen students that have done all of these. Okay? But from a personal point of view, is the sort of more nebulous one. But you will have taken in a PhD you have owned a project that nobody else knew how to make work. You have solved all these different problems and you managed to get it right the way to completion. You've achieved something, you've gone into the unknown and you've solved something. And when you do that, the skills that you build and what you've demonstrated, problem solving, resilience, how to work as a team with people, courage, you've gone into the unknown and you've solved it. And no one will ever be able to question those life skills. No one ever looks at someone with a PhD and says, can you really solve things? Or can you really keep going when times get tough? It's like, yeah, of course you have. Like, that's what you did. And nobody ever questions those. And you will know carrying forward in life, you did that. And it's an amazing thing to have behind you. So summing up and kind of taking you back to where we started, this is an absolutely beautiful, transformative time to be doing biology. You're coming to an amazing university, and the possibilities are literally just opening up in every direction. It's a great time. Please come and talk to us more if you need to know more information, and it would be lovely to see you here at U of T. Thanks very much.